It's my pleasure to introduce Jocelyn Holt. Jocelyn has been a close colleague of mine since we founded Aggie Women in Entomology years and years ago as graduate students. So Jocelyn is currently a NIFA USDA pre-doctoral fellow at Texas A&M University. She completed a bachelor's degree in biology in Cal State Polytechnic University, and then went on to complete her master's degree in biology at Cal State Northridge. She's currently a PhD candidate mentored by Raul Medina. She works on genetics and microbial communities, influence mutualisms, and facilitate invasive species establishment. Her current research investigations focus on bacterial composition and population genetics of two invasive pests, the sorghum aphid and the tawny crazy ant. And then I also wanted to mention that throughout her entire career, Jocelyn has been an active participant in diversity, equity, and inclusion initiatives, both at the university level and also at the societal level.
Yes, thank you so much for that wonderful
I was muted. I wasn't finished. When did the host muted me? Yeah, I have. Where did I get to?
You were all the way to the end. I think you were just. It sounds like you were just going to introduce Jocelyn. So you're good.
And then, well, the last thing I have scripted a thing. So and the last thing I wanted to mention, in 2019, Jocelyn was the recipient of the Comstock Award. And I don't remember where I was. So Jocelyn, I'll hand it over to you.
Thank you so much, Chloe, for that wonderful introduction. I really appreciate that. I'm very excited today to talk with everyone about my research on uncovering the secret life of sorghum aphids through genetic and microbial analysis. And before I start talking about my research, I would like to share just a little bit of background about myself. So I grew up in Southern California, which is a biodiversity hotspot. And in this state, I had access to mountains, deserts, and ocean. And along with that, there's lots of plant diversity and insect diversity. And I really think that this is where I developed my passion for studying insects and the interactions that these critters engage in. And so now everywhere that I go, I'm looking for insects through my field work and hiking, and I'm also photographing them because I think they're amazing and they're beautiful. I'm also a proponent of entomophagy, so the consumption of insects. I think that if we eat insects in our processed foods anyway, we have all those bug bits, then we might as well dive in and eat some whole bugs. So here at the top, you see that there are some palm weevil grubs that taste like bacon, and I've also eaten ants when I was in Ecuador, and these are lemon ants. And so because of the acid that these ants produce, they do taste very lemony. I taught before pursuing my graduate career in entomology at San Jacinto College North in Houston, Texas. And this was a fantastic experience because it's an Aspen college that serves a very large Hispanic and Latino population. So you get many underrepresented students who are interested in pursuing STEM. And really, as being like a Latina in STEM, that allowed me to encourage and support the students that came through my classes. So not only did I get to share my excitement and my enthusiasm for biology and insect interactions, but I also got to encourage them and let them know that yes, they belong in STEM, and there are careers available if you choose to pursue biology or entomology. And so my teaching background, because of my experience at San Jacinto College, means that I've taught face-to-face and distance learning classes as instructor of record. I've been able to recruit undergraduate student researchers to participate in research projects. I've had opportunities to guest lecture in integrated pest management insects in biotechnology, insects in society. And so this has given me lots of opportunities to connect with students, to share my enthusiasm for entomology, and also to show them that there are lots of opportunities for them to be in entomology and for them to get careers where they can be successful. And I do carry this commitment to like encouragement and mentorship into my research areas as well. So I've had opportunities to either informally or formally mentor undergraduate students and graduate students. I'm also, for the past 4 years, have been a participant in Launch at Texas A&M as a panel member to help provide guidance and information to graduate students who are taking on undergraduate student researchers, and really to help them tailor their approaches to best promote success of those students. Also, I am currently an ESA Entomentos mentor in the mentorship program, where I'm mentoring a first-year PhD student as she's developing her research project. So all of these things to me are something that I think are very important, and I'm glad that I get to participate in to help bring through that next generation of entomologists. And it's really important because, when you are underrepresented in STEM, or you come from a diverse background, or you're a person of color, having that tailored mentorship is something that can really help you feel like you belong and promote your overall success. Yes, that person who's being mentored is going to go through and do the hard work and have that determination, persistence, and grit. But having that support network is something that makes you feel like you belong. And by doing that, I think that not only can we recruit diverse students into STEM, and particularly in entomology, but we can definitely retain them. And this is also something that I've done at the university level, as Chloe mentioned earlier, as a founding member with Chloe in Aggie Women in Entomology, where we were able to promote women and diversity in STEM. And I think one of the exciting things about this, in addition to the women seminar speakers we invited, the outreach we did, the seminars that we coordinated, the panels that we hosted for undergraduate students who are interested in going to graduate schools, is that this network really provided a great support and foundation for its members, to the point where we've had such great success from the people who have been participants of this. We have 2 assistant professors from this group. We have a postdoc, we have a consultant now for a company, and then we have me, who, I've been very excited to receive my USDA NIFA grant that I wrote, but also I'm looking forward to that next adventure in my career. And I've also, participated in service through the society at ENTSOC, ESA, the Entomological Society of America, where that role in leadership as Student Affairs Committee Chair has really helped me form those networks and connections, and be able to participate in discussions that are focused on diversity, equity, and inclusion. So even though I may not be officially on committees for these things, I do find opportunities to discuss this and to talk with members of some committees, so that we can help make our society a more welcoming and inclusive place. And just as a quick plug, if you are attending ESA in person or remotely, my colleagues and I, including Chloe, Estelle Martin, and Karen Poe, are co-organizing a symposium titled Celebrating Entomological Diversity While Advancing the Management of Invasive Species. And that's going to be held Sunday, the 31st of October at 8 a.m., but content will also be available online as well. So now that I've given you a little bit of overview of myself and my previous experience, I want to shift gears and talk about my research in genetics, in assessing microbial composition, and seeing how that modulates symbiotic interactions, and in particular, mutualisms, are these beneficial interactions. And the research that I'm going to talk about today is focused primarily on these two invasive pest insects, sorghum aphids and tawny crazy ants. So I'm going to go through and talk about the sugarcane aphid microbiota, and then move to sugarcane and sorghum aphid genetics, talk about sorghum aphid and tawny crazy ant interactions, as well as some ongoing research and my future vision. So why should we care about microbial symbiotic interactions? I think that if we take a step back and look at ourselves as people first, that this may help us understand the important role that these microbes can play in insects and why it's worth investigating. So in 2007, the Human Microbiome Project is something that positively influenced microbial research. So by having this research that then opened up a whole host of possibilities, understanding the microbes that are in our body, that in actuality, we think of ourselves as like one individual, but we are really a community of organisms. So we're maybe somewhere around an estimate of 57% microbial cells and 43% of our own cells. So we're not just individuals, we're one entire community that functions together. And this research also then made microbial research much more accessible to other fields through high throughput sequencing, because it became more common, and then costs for sequencing this type of research decreased. Now, when we think of ourselves, it's important to note that microbes can influence our health and behavior. And as one example, right, we're kind of getting in that stressful time of the year with ESA approaching, and then grading and finals and all those things. So, you know, we need to remember to feed our gut microbes, because our gut microbes, all those bacteria, produce these chemicals or neurotransmitters that then secrete, you know, this dopamine and serotonin, which can travel up the vagus nerve and change like how we feel and how we may respond to our surrounding environment. And so really, if we want to best promote our health and have the most possible outcome for being healthy and happy, we should be focusing on feeding our gut microbes and feeding them well. So knowing that about ourselves, now that we're really like this whole community of organisms, of our cells and microbial cells, we can take this information and transfer it over and think that really when invasive species arrive to a new location, they do not arrive alone, right? So it's not just that one insect, it's not just that propagule, it is that individual, whatever genetic composition it is, so the genotype, plus the whole microbial community that it harbors both inside and outside of itself. And so given this, now we can start to look at how microbes can influence host resource use. And we're gonna see an example first with kudzu bugs, because kudzu bugs invaded the continental US relatively recently. And when they first invaded, it was thought that maybe it would not be a pest on soybean. However, kudzu bugs that are pests on soybeans have a particular strain of bacterial symbiont, this Candidatus ischiquala capsulata. And because these symbionts are acquired by the offspring through symbiont capsules that they can consume, it is possible that they can acquire other forms of this symbiont. So if you take the pest strain of Candidatus ischiquala capsulata from kudzu bugs that consume soybean, and you give it to a non-pest that consumes kudzu, now suddenly you can have a new pest that is capable of consuming soybean. So just by changing the strain of microbe that this insect has, now you get a new pest. And we can take that research that was done by Hoskow and Brown, and we can look and ask, well, how might this apply to some of the things we're seeing with sugarcane aphids? And sugarcane aphids have been a pest in the continental US since 1977 in Florida on sugarcane. And then it wasn't until 1999 that we had reports of sugarcane aphids on sugarcane in Louisiana. And then we had some more time go by, and suddenly in 2013, both in Texas sorghum and in Louisiana sorghum, you had reports of sugarcane aphids being a pest in this agricultural crop, and it was devastating. Growers had anywhere from like 50 to 100% yield loss because this insect was so damaging that first year. And now, if we're thinking about the geographic distribution of these aphids on sorghum, it has migrated from Texas and Louisiana and spread both to the West Coast and to the East Coast. So it can be found all the way from California over to Florida, where you have sorghum production. And so if we think back to the example of kudzu bugs, we can ask, are bacterial symbionts something that may be responsible for the aphid pest outbreak we saw in 2013 and this possible host shift from sugarcane to grain sorghum? And so to do this, my colleagues and I had aphid samples that were collected from sugarcane. So those are the reddish diamonds, and also sorghum, which are the blue circles. And they were collected from Texas, Louisiana, Alabama, and Florida. And we also had some samples before the 2013 pest outbreak. So these are samples collected from 2007 to 2009 on Louisiana sugarcane. Thankfully, because Samuel Nabush and his research team had samples from this period of time that they saved. And then as a comparison, we also had samples from South Africa of aphids that fed on sorghum, because it's been a pest there for over 30 years. So to examine this microbial composition, we took a two-pronged approach, and this was using diagnostic PCR that was done by the Jen White Lab at University of Kentucky, where screening for known bacteria was done. And this is something, in aphids that you might expect to see: Haemotinella defensa, Arsenophilus, Rickettsia. So these are the types of bacteria that were screened for. And then I also submitted sequences for high throughput sequencing for the V3, V4, 16S ribosomal RNA gene region. And this was analyzed using CHIME and UParse for OTU analysis. And because of time constraints, I'm going to show you just the high throughput sequencing data, but I will say that the diagnostic PCR matched up with these findings. And as a quick spoiler alert, there are differences in microbiota between aphids that feed on grain sorghum and aphids that feed on sugarcane. So when we look at these microbial differences, first, I'd like to point out that we do not see the facultative symbionts, so those symbionts that may be present in aphids but aren't essential for aphid survival that we might expect. So we don't see Arsenophilus, we don't see Haemotinella defensa in the samples we collected, and we do not see Rickettsia. However, we do have some bacteria that maybe we didn't entirely expect to find in aphids that are significantly different. So we have Archobacter, Bacillus, Citrobacter, and Flavobacterium. And I think that it's very interesting that we found Citrobacter, because in fruit flies, so tephritid fruit flies, the bacteria Citrobacter is known to help it detoxify insecticide. So in California, these tephritid fruit flies are a major pest on fruits. And by having that bacteria, having insecticide treatments is not effective and requires other management approaches. So, I think it's certainly an avenue for further investigation to see what potential role Citrobacter may play in these aphids. While there are differences in the microbial abundances between aphids on sugarcane and aphids on grain sorghum, there is not like one particular bacteria that stood out as being present or absent in either one. So based upon this data, I'm going to say that the bacterial symbiont composition was not something that caused a host plant shift. And this then leads to like further questions and further investigations into what could have been the reason for this. And this requires then looking at invasive insect genetic composition and how that is associated with host use. So we're going to take an example. This is a classic example of pea aphids. And pea aphids have what we call host-associated differentiation. So this means that they're all the same species of pea aphid as of this study, that genetically distinct pea aphids are better at using different resources. And in this case, that resource is a host plant. So they're all legumes. And if we look at the visual at the bottom, we're gonna focus on two different genotypes of pea aphid, the genotype in gray, which is A, and the genotype in blue, which is G. So you see represented by the colors that they are genetically different from one another. And if you look below, the flowers of those host plants are also different. So the genetics correspond to the difference in host plant use. And then this led me to ask, is the pest outbreak a result of the introduction of sugarcane aphids with host-associated differentiation? So did we maybe get the introduction of this sorghum-adapted aphid that then is responsible for this big pest outbreak we saw in 2013? And in order to answer this, my colleagues and I collected aphids from across the continental US. So now this also includes samples in California, where these aphids can now be found, as well as Mexico and in the Caribbean, and on both sugarcane plants and sorghum plants, and then like a corn plant in maize. And looking at the aphid genetic composition, both using mitochondrial cytochrome oxidase 1 and microsatellites, what we see is there is host-associated differentiation. So at the bottom, that red box, which represents haplotype 3 of that CO1, we have multi-locus lineage D, so aphids that are feeding on sugarcane that occurred before that 2013 pest outbreak. And then if we go over, it's kind of slightly further away and genetically different. We have the gold box, which is haplotype 1, and we see in that yellow circle, you have multi-locus lineage F, and these are the aphids that are reported during that 2013 pest outbreak and are sorghum adapted. So based upon this information, it does appear, yes, that we have host-associated differentiation of like this one sugarcane aphid species. However, if we start to look further into this, because like there are kind of like big differences in how far apart those haplotypes are, we can ask, well, are the aphids on sorghum actually the same aphid, or maybe are they this cryptic aphid species because they're small and looking with the naked eye, sometimes they're hard to discern. So to do this, my collaborators and I had aphid samples that were collected globally, both on sugarcane and on sorghum. And then morphometric measurements were done in order to assess if these aphids are actually all sugarcane aphids, or if some of these aphids maybe are sorghum aphids, and these are two distinct already classified species in the literature. And assessing differences between these two species means looking at the ratio of the process terminalis, so the tip of the antenna, the hind tibia, the cauda, and the siphonunculi, which are sometimes called cornicles. And when those differences in ratios are looked at, so the process terminalis and the cauda, we see there are significant differences in those aphids. So those that are going to be sugarcane aphids and those that are sorghum aphids, we see significant differences between the hind tibia and process terminalis ratios. And we see significant differences between the process terminalis and siphonunculi. And so all of this then leads to the fact that actually we have two distinct species. So we have Melanophis saccharine on sugarcane. Those are those samples from Louisiana and other locations globally. And then we have Melanophis sorghum on sorghum. And this can further be supported with some molecular analysis. So remember I mentioned that there are differences in CO1 haplotype and also those multi-locus lineages, and looking at elongation factor 1 alpha. There's also a mutation that helps to very clearly discern these sugarcane aphids and these sorghum aphids. So that mutation is a T for sugarcane aphids and an A for sorghum aphids. So, when we're looking at invasive insects, given all this information, sometimes we really need to ask, is the thing that we're looking at really the critter that we're interested in? Because when we're talking about aphids or other small insects, sometimes they're very difficult to discern, and it takes, as we've seen, multiple avenues of information and support to actually tease apart when we have different species that are very difficult to tell apart. So that can include those microbial differences, the host plant preference differences, the genetic differences, and those morphometric differences. And so when we're thinking of pest management, I think it's important to keep in mind that complex challenges require integrative and complex solutions. So oftentimes with invasive insects, the first mode of approach is to spray widely to kill the invasive critter. And while this can work in some instances, we saw with work done by a different lab group into fruited fruit flies, there's a bacteria that allows this fruit fly to detoxify insecticides. So that may not work, and it may actually promote this pesticide treadmill for other insects in that agroecosystem. And that's something that we don't want, because we want effective approaches for pest management. And so very exciting, and this recently, semi-published in release review paper that I did with my colleague. We talked about really looking at ways for tailored and sustainable pest management. So I like to think of this as being akin to, say, personalized medicine, where you have an individual and the doctor is looking at that person's genetic composition and chemical composition to make sure that the plan for treatment best complements those factors. And so when we're thinking of pest management, I think it's important to kind of take that perspective.
Can anybody hear Jocelyn?
No, I was just gonna say, I just text her. I'm gonna call her.
I thought it was just me.
All right. So, when we think about pest management, we should really think of it similar to having like precision health, right? Like personalized, like tailored medicine. Because when we have personalized medicine, that takes into account a person's genetics, that person's chemical composition, and how that may interact with chemicals that they may be taking, medications, or how their body may react to certain treatments. And so really then when we're thinking of pest management, it should be tailored and it should be more like this precision agriculture. And in order to be able to do this, we need to incorporate both genetic and microbial analyses to obtain sustainable pest management. And we have so much exciting novel technology that's coming out and can be applied into agriculture. So with the advent of gene editing that was developed by Jennifer Doudna and Emmanuel Charpentier, you have this ability to have like precision editing of pests or even microbes that can then reduce abundances or reduce the ability to vector or like transmit pathogens. So that's something that's very, very exciting. And we can also incorporate artificial intelligence and drones into pest management. So thinking about using this to survey for pest damage or for invasive insects, and also to use maybe some targeted treatment of either biopesticides or insecticides. This may help reduce off-target effects. And we can also incorporate the use of environmental DNA and soil microbes into pest management. So in aqueous solutions, so in aquatic ecosystems, this is something that is done to survey the diversity of organisms there or detect invasive species. But I know that a lot of research has been done through the Fonseca lab here at Rutgers, looking at incorporating the use of environmental DNA into these terrestrial ecosystems. And I think that's very exciting because yes, you can identify pests through scouting, but by looking at environmental DNA, this may help us detect these insects before we can ever see them. And also by paying attention to soil microbial communities, how we can promote the overall health of these soil microbial communities, and maybe even make synthetic soil microbial communities that can better promote plant health and reduce damage of invasive pests. So when we're thinking about all of these things, we should think of microbes, the genetics of the microbe, the genetics of the host organism, and how all of this interacts to like modulate these symbiotic interactions. And we can add layers of complexity when we talk about invasive species, because when they arrive to these new invaded locations, they can establish novel symbiotic interactions. So here we have tawny crazy ants, which is an invasive ant, and sorghum aphids, also something that have not interacted previously in their native ranges. And what we see is that the ants will go and tap the aphids with their antennae. So they're tending these aphids. And it's very much like people would tend cows or goats to be able to get milk, which can be a very nutritious substance. So these ants tend the aphids. They get honeydew, which is this rich carbohydrate that can then help promote colony growth. And this is something of concern because tawny crazy ants can quickly establish new mutualisms. So in Colombia, tawny crazy ants were introduced to help control leafcutter ants, which can devastate agricultural crops by removing lots of pieces of leaves, and also to control snakes. And when they were introduced, these ants tended mealybugs in grasslands, which increased phloem consumption, resulting in the drying of the grasslands and the reduction of forage quality. So this is something that is very negative towards those who are looking to graze cattle. And in Florida, there are already reports of tawny crazy ants being opportunistic tenders. So what does that mean? They tend a variety of different hemipterans. So that includes whitefly, aphids, scale, and mealybugs. And if we look at the red boxes, what we see is that those particular hemipterans are invasive. So there's already a precedence of this invasive ant establishing these new symbiotic interactions with invasive hemipterans. And when I first started thinking about this research, this is something that some colleagues said would not happen because sorghum aphids are known to flick honeydew during excretion. So when we look at this video at the yellow arrow, what we see is that the sorghum aphid is going to slowly rear away from the plant. And you see this little droplet of honeydew develop, and then it gets flicked away. So this prevents the honeydew from accumulating on the area around the aphid and reduces the likelihood of fungal growth that could then kill the aphid. However, what I've found is that aphids modify their honey excreting behavior in the presence of tending ants. So here we have tawny crazy ants that are going and antenating these aphids, and you'll see that the aphid then rears away from the plant and releases honeydew to the ant then consumes. So this is something that is very exciting because it means that even when we see a particular behavior in invasive species, that does not mean that that behavior may not be modified as it encounters other insects or other organisms. And so this led me to ask, does the interaction between ants and aphids increase aphid biomass or increase plant chlorosis? And to do this, I wrote a grant through the Texas Ecological Laboratory to be able to have supplies and funding for those supplies, as well as to access private property to collect some of these aphids. And for data collection, I had a treatment and a control. I've put the mesh cage down on the treatment so that we can see how it's set up. We have 20 reproductive female aphids that were placed in there, 500 workers and 1 queen. And I should say that these particular aphids are fully parthenogenetic. So that means that you have a reproductive female and she has lots of clones. So it makes it very easy to work with this aphid. And I recorded the number of aphids at an observation site, the number of antenating events, the number of ants observed tending. And this was all done using the Boris software for 10-minute observations over 5 days. And while I did collect all that data and the control just has the 20 reproductive females as a comparison, I'm just going to show some of the biomass data. So ant tending did not increase aphid biomass. You see that there are some differences in aphids in the presence of ants, but they're not significant differences. And research done by my mentee, Jose Torres, did not find an increase in sorghum leaf chlorosis in the presence of ants, but you do see that there is a trend towards there being more chlorosis. So, you know, this is a 14-day experiment, but it's something that we should still be aware of and continue to look for. So I think that this data is exciting because it provides a foundation for this ant and aphid interaction, this change in behavior from flicking to presenting honeydew to the ants. And it's something that we should still keep monitoring because tawny crazy ants interact with these sorghum aphids on invasive Johnson grass, both in agroecosystems and also in these more natural ecosystems. So over time, or maybe like observing greater observations, we may find that there is an increase in biomass, or we may find that there is an increase in this leaf chlorosis. And so, I think that this story shows that it's important to consider the interactions that invasive species establish once they invade a new location, because although sorghum aphids and tawny crazy ants had not ever interacted before, this is a new symbiotic interaction that has developed relatively quickly and is something that could be of both agricultural and natural ecosystem concern in the future. And we can continue to tease apart some of this complexity by assessing the microbial composition of aphid honeydew. And you may be wondering, aphid honeydew may be so important in attracting ants. And in a study done by Fisher and colleagues, the bacteria Staphylococcus psilocybe is something that is in black bean aphid honeydew. And this is something that is very attractive to ant foragers, or these black garden ants. So that then led me to ask, does sorghum aphid honeydew microbial composition vary by host plant or across time? First, what's in this honeydew, all the shiny splotches we see on this sorghum leaf, and then how does this vary, and how might that impact how it's attractive to ants? So to do this, my colleagues and I had pre-weighed discs for honeydew collection. And then we had aphid clones that were reared in clip cages. So there were 10 clones per treatment. We had 20 plants of either grain sorghum or Johnson grass. And then on each of these plants, there were 3 different clip cages for honeydew that was collected.
Did we lose her again?
Yeah, let me call her.
Okay.
Okay. So honeydew was collected at 2472 and 120 hours from these clip cages, and also at 7 days from samples in the greenhouse. Genomic material was extracted with a DNA power soil kit, and then this was sequenced for the V416S ribosomal RNA gene region, and it was analyzed with the DATA2 pipeline. And what we found when we look at this microbial composition is that visualizing it with non-metric multidimensional scaling, we don't see these significant differences. And this is supported with a Permanova analysis. So samples, whether they're in grain sorghum and Johnson grass, appear to be very similar. We can look at this microbial composition across time, and there are significant differences at these time points, and doing analysis for individual bacteria, so looking at like a linear analysis, what we see is that we do have differences in Buchnera, Herbaspirillum, Ralstonia, and then a presence of Staphylococcus, and then Corneobacterium pops up, but it's something that maybe we need more samples of to really assess what's going on there. So if we compare this between honeydew and the whole aphid, really the only bacteria we have in common is Buchnera. And this may be because the aphids are parthenogenetic. So maybe some of those offspring are like destroyed inside the aphid, and then that buchnera gets excreted along with the honeydew. And then the other two interesting bacteria are that Ralstonia is present, and in other plants, Ralstonia is known to be a plant pathogen. And we also have Staphylococcus. So that's very exciting because remember, with the black bean aphids, Staphylococcus psilocys in honeydew was very attractive to ant foragers. And work done by my colleague, Chris Wright, looked at the attractiveness of grain sorghum honeydew versus water to a parasitoid wasp, Aphelinus nigritis. And what she found is that this honeydew is very attractive to parasitoids, much more so than to water. So there's certainly something in here that is attractive to parasitoids. And I would then likely think that there's something very attractive in this honeydew to ants. And kind of as a next step for this, I think that, taking those same samples at different time points and submitting them for fungal composition of the honeydew is very exciting, because right now, what I've talked about really is the bacterial composition in aphids and in honeydew. But we have the development of sooty mold on leaf surfaces in the presence of honeydew. So finding out what's already there and what develops across time is something that I find very interesting, and I'm looking forward to getting started next. So I'm going to just ask a quick question. Since we've had some technical difficulties, would you like me to continue talking about upcoming research, or you want me to go ahead and stop here and we can just wrap up?
I'd say go right ahead.
Okay, great, thank you. Thank you. So I'm really excited to share some of my upcoming research and some my future visions for research. Still focused on genetics and microbes and invasive species. So in 2020, I was awarded my USDA NEFA grant, and this is exciting because while this is a large-scale genomics project, I think that by assessing potential differences among sorghum aphids, right? So we know now it's classified from sugarcane to sorghum aphids occurring on grain sorghum, that maybe if one day a producer can easily identify the genetics of these sorghum aphids in their field, so I'm talking maybe developing quick screening kits so they know what type of pests they have, if maybe some are more resistant to insecticide than others, then they'll know how to better fight them and will have less economic loss. So that's something that has very exciting implications. And in order to do this, we need to incorporate fine scale molecular markers to detect genetic differences. And I'm just going to present an example involving the resolution differences between using fewer molecular markers and then thousands of molecular markers. So in tawny crazy ants, this is work done by Error and colleagues, the use of broad scale molecular markers detected genetic differentiation in the native range, but not in the introduced or invasive range. So 13 microsatellite markers were used, and the ants throughout the continental US all appear very homogeneous. However, when I use fine scale molecular markers, so now we're talking about thousands of molecular markers, 24,341 SNPs, or single nucleotide polymorphisms. What I find is there is population genetic structure in tawny crazy ants that is associated with geographic range. So yes, there are some differences in the native range. And then if we look in the invaded range, it's grouped by geographic region. So that includes Florida, and then Louisiana, Georgia, and Mississippi are kind of grouped together, Alabama, and then Texas. And I'm currently just assessing using structure what the subpopulation structure may be among the ants in the US, because it's entirely possible that the samples from Argentina, Colombia, and Peru were so different that it kind of masked some of those differences in the analysis with all the samples together. And so now with my current USDA NEFA grant funding, I'm assessing whether there are fine scale population genetic differences among sorghum aphids. So using CO1 and microsatellite markers, my colleagues and I found like there are some differences, but there seems to be like one big superclone in the US when it comes to aphids on sorghum. And work previously done in the Medina lab using amplified fragment length polymorphisms did find, that's hundreds of markers, did find that there are some population genetic differences. So I think that as we increase these molecular markers, there can be a greater resolution, and I would expect to find some genetic differences among these aphids. And I'm also then with the same USDA NIFA grant looking at corresponding fine scale population genetic structure among its obligate symbiont Buchnera aphidicola. So here we have that abdomen of the aphid in blue, and then the Buchnera contained in these areas that are shown in green. So I think that if I find that there are some population genetic structure differences in the aphid, then it's entirely possible that there will be corresponding differences in Buchnera. And also then that will allow me to further look at Buchnera and ask, like, are there any differences that maybe like ecologically facilitate this aphid to be a pest? So in other studies, there's a mutation in Buchnera that allows for greater heat tolerance, which means these aphids can expand into more geographic regions. So maybe something like that is happening. And if that is, then that might be why we see it being able to occur from both the West Coast all the way to the East Coast. I'd also like to continue my research in assessing microbial symbiont communities and their potential roles. And so remember that with tawny crazy ants, I found fine scale population genetic structure using these thousands of molecular markers. And so the next thing I want to do is ask, does the microbial community composition differ among these genetically distinct tawny crazy ants? This is work that's currently funded with my Texas Ecological Research Grant. So I see my research as being something that's very collaborative, and that is something that takes place with a lot of invasive species in these agroecosystems. But I also am working towards comparisons with these invasive species and more of these natural ecosystems. So what types of differences do we see? And this work is something that necessitates collaboration, because being able to collect samples from such a wide range of ecosystems and globally means that you absolutely have to collaborate to ask those big questions of what's actually going on. And I know that you do have lots of brown marmorated stink bugs over in New Jersey, and we also now have them here in Texas. So I think it would be exciting to collaborate potentially on some research, looking at characterizing the attractiveness of stink bug secretions to ants, as well as that microbial composition of those stink bug secretions. And I think that this is something that could be potentially funded through an NSF, Understanding the Rules of Life, Microbiome Interactions and Mechanisms, or an NSF Integrative Research in Biology grant. I also think that it would be very interesting to assess genomic differences between native and invasive cactus moths. And this is something that, invasive cactus moths are very damaging to Opuntia. So understanding what those genetic differences is may help us in the future with some pest management. And this could be something that's potentially funded through an NSF Integrative Research in Biology grant or an NSF NIFA Plant Biotics Interactions Program grant. So, I hope I've shown that my research on genetics and microbes and how this can modulate symbiotic interactions and in particular mutualisms, even though it's focused currently on sorghum aphids and tawny crazy ants, is something that I'd like to continue to extend into other invasive insect systems, and both in agroecosystems and in natural systems. And, I think that as we continue to listen to the interactions of insects and organisms around us, as well as to uplift diverse voices and the insight of diverse individuals, that we're going to make great strides both in STEM and in entomology. So thank you so much. And I will take any questions that you may have.
Thank you very much. Are there
Yeah.
any questions for our speaker?
And I don't know how to put the screen back. So
You should be able up at the top, hit
I hit it.
and you should be able to unshare.
Okay, let me see if I can end the show and unshare. Where did Zoom go? Okay, hold on.
I see. Okay.
Oh, okay.
Yep. There you go.
Oh, there we go.
Yep.
Oh.
Perfect. Any questions? Chime in. Just unmute yourself.
I don't have a question, but that was great.
Oh, thank
Really
you.
good.
I appreciate that. Yeah. And I appreciate everyone like working with me through all of these technological difficulties.
No, not a problem. All right. Well, I guess if we have no questions, we'll move to the class part of this. And I'll ask everybody to who's not in the class to leave the session, and we'll continue on.
Okay, perfect. Yeah, so I just stay here then?
Yes. Yes.
Okay, great. Thank you. Oh, here we go. Let me see.